I warmly welcome all the participants. Today's webinar of genetics and genomics in forest improvement. Our today's speaker is Professor Dr. Matthias Lodem. He is from Pune Institute of Forest Genetics from Grosshansdorf in Germany. Before I hand over the mic to Dr. Matthias, I will request Professor Dr. Khan Habibullah Khan, our director, for some opening remarks.
Thank you, Dr. Ikrar. On behalf of Center for Advanced Studies in Agriculture and Food Security, I warmly welcome very distinguished speaker today, which is Professor Matthias. My dear colleagues and students, I am really very hopeful that you will get benefit out of this very important webinar. We have very severe issues of trees like citrus greening, like tree dieback. So these are very gigantic issues in Pakistan, especially in the current climate change scenario. So their genetics and their genomics are very much important to dissect the issues, to have some counter actions to mitigate these issues. So on behalf of the center as well as University of Agriculture, thank you very much for accepting Professor Matthias to share your experience on tree science, especially genetics and genomics. Over to you, Professor.
Yeah, thank you very much. First of all, do you hear me?
Yes.
Yes, fine. That's good. That's the first. So, first of all, I would like to thank you for the kind introduction, Professor Sultan Khan, and also for the kind invitation from Dr. Ahmad Rana. I'm happy to share with you some of our work which we are dealing in institutes to present some of try to share my screen. Do you see the screen? The presentation.
I can see.
Okay, fine. So the topic of the talk today is genetics and genomics in forestry improvement. But before I'm going into this topic, I would like to introduce shortly the Thünen Institute, where I am belonging to. The Thünen Institute is in Germany. Yes, it's a research institute within the scope of the German Ministry of Food and Agriculture. It was established in 1st of January 2008 during the reorganization of the departmental research of the German ministry. The headquarters of the Thünen Institute is located in Braunschweig. So I'm very sorry. I tried now to connect my mouse because I've seen. So do you know why it's not showing? Yeah, okay. I think it's white. Okay, that's a disadvantage. Okay, maybe you can see it. Braunschweig here in the picture is in the middle. It's more or less north of Germany, but in the picture here in the middle, in the lower part of this picture, maybe you can see it. It's an independent higher federal authority. It's organized as a presidential system, and the field of activities are advice for policy, but also research. The institute I am working is the Institute of Forest Genetics, the European Institute of Forest Genetics. It's located in Groß-Hansdorf. That is the main part of the institute, but also we have a small branch office also in Bad Sieversdorf. Bad Sieversdorf is located in the east of Berlin. Maybe you can see it on the right side of the picture. Good. As I already mentioned, we have two locations of the institute in Groß-Hansdorf and in Bad Sieversdorf. I'm working in Groß-Hansdorf. The director of the institute is Bernd Degen, and I'm the deputy director of the institute. The infrastructure of our institute is very huge. We have more than 2,000 square meters of land to 8 weeks. The regeneration of new shoots, which can be cut from the middle picture and can be rooted, and you find it in the white picture, can be rooted, and and then we can harvest the leaves and the stems and use biotechnological methods. It's easy to transform regarding genetic transformation. This is a very big advantage for many poplar clones. The genome size of poplar is relatively small. It's just 3 to 5 times larger than Arabidopsis. It's about 1/50 of the pine genome to get an impression about the small genome size of poplar. Genome sequence has been published by Jerry Tuskin already in the year 2006. The genome consists of about 42,000 loci, nearly 43,000 loci containing protein coding transcripts. The genome is publicly available in Phytosome, and I have checked texture, stem, growing root, and capacity of regrowth from rootstock. Techniques for tree cross-pollination systems. It will present you just 3 examples of approaches which we have done in our lab. First, we have used the gene PCBER1. This is a gene in the lignan biosynthesis. Lignans are secondary compounds of for plant defense. Lignans have also a therapeutic relevance against cancer and further diseases. And here we have started with first an overexpression approach. That means putting the gene PCBER under the 35S promoter and overexpress this in poplar. We have obtained several transgenic poplar lines, but all the independent transgenic poplar lines, as you can see in the figure on the left hand bottom, you can see the columns, and there is no difference in plant height, neither in the first year, it's the smaller columns, but nor in the third year. So the overexpression doesn't have any phenotypic, doesn't show any phenotypic variation, and also the analysis of the book. There is more or less similar between the transgenic lines and controls. All this work has been published in 2019 in Planta by Tobias Brookman and co-workers. The next point is that we use the same gene, but not to overexpress it, but to knock down. And here, the knockdown approach showed a significantly increase of the height in 6 out of 9 lines. The increase was about up to 38%. No significantly modified wood composition. expression and also the lignan content also didn't significantly change. Here in this slide I show that this is currently determined, but later on we didn't find any variation. And in the slide on the right hand side you can see the three different transgenic lines, and in each picture on the right hand you have the control line, which is much smaller than the plants in which the gene PCBAR1 has been knocked down. So, in summary, PCBAR1 is a promising gene for increasing biomass, and lignans, but this is not as important, valuable approach for pharmaceutics. A second example is changing the cytokinin status in poplar plants. Cytokinins control different meristematic activities. We have chosen a gene which is called repressor of cytokinin deficiency, or short ROC gene, and we obtained different transgenic lines which show increased plant height and stem diameter. As you can see in the picture, on the left hand side the wild type, and then three independent transgenic lines. The differences are not so huge as we have found it in the knockdown approach of the gene PCBAR1, but it was significantly increased, the height. Also, this work has been published this year in the International Journal of Molecular Science. When you are interested in, please refer to Michael Riefler and co-workers and finding details of this research. And finally, also here, this is seems to be complicated picture, but isn't this? Is not. It's a work on the gibberellins and the overexpression of the GA20 oxidase genes, which has been first published by Erickson and co-workers in Nature Biotechnology. We have used this gene under the expression control of the 35S promoter. We found, similar to Erickson, an increase of plant height, but also thinner leaves, and thinner leaves are a disadvantage for the stability of plants. So we replaced the 35S promoter with a selenium-specific promoter, which has been also been done by Jeon and co-workers, published in Plant Biotechnology, and also in our lines, as you can see here from the different graphs and columns, the transgenic lines overexpressing the GA20 oxidase gene are higher, and when under control of the DX15 promoter, the selenium-specific promoter, the stem diameter are more or less similar like the controls. So, coming to the next topic, the fast breeding technology. Here, it's let's say on the left-hand side, in comparison to natural variants which show early flowering, at least in the Populus alba clone 6K10, which has been published by Rick Malin already in 2004. But also, you can see on the upper slides, on the left-hand side, in Norway spruce, P.C. avis, there's a mutation called, this is a dominant mutation, Acrocona, and as you can see here from the 5-year-old plant, that many cones have been developed, which is absolutely unusual in Norway spruce, because Norway spruce show the first cone formation after 10 to 15 years. Using a transgenic approach, you find it on the right hand side, first with this leafy gene, 35S leafy. Leafy is a meristem identification gene, and putting this gene under the 35S promoter transform male and female poplar, and you can see that this gene is able to induce early flowering already in the in vitro culture. And also we can see it in the greenhouse, as you can see in the lower picture. The disadvantage of this gene is that instead of catkins, which are normally produced by poplar, just single flowers are produced, as you can see in the female with the red arrow, but also in the male clones, also indicated by a red arrow. So single flowers instead of catkins are not so a good system to work with, and therefore we decided to use the early flowering gene, flowering lupus T, which has also been tested in several species, and we have put this gene under either the 35S promoter, but also under a heat-inducible promoter, and especially by using the heat-inducible promoter, controlling the flowering lupus T or short FT, we are able, after putting in heat shock, that also the plant started to flower in the in vitro culture, as you can see in the middle picture, but also in the growth chamber after 8 months. Here you can see that not just single flowers have been developed, but also complete catkins. And this approach is promising, so in the next step we have analyzed and tested whether these catkins induced by the FT gene under the heat-inducible promoter are fertile, and we have tested different light conditions, short day and long day conditions, and warm and cold treatments, and all this work has already been published by Hans Hönnecker and co-workers in Tree Technology in 2016. When you are interested in the details of this work, there you can find additional information here. We have performed successfully crosses with both male and female flowering, but to summarize shortly this work and to introduce the high speeding technology, temperature and not photo period is the key factor required for the development of fertile flowers in early flowering FT transgenic poplar. And for the first time, crosses were successfully performed with male and also female early flowering lines. The non-reproductive phase, usually in poplar, starting from 7 to 10 years, can now be shortened to 6 to 10 months, and fertile flowers can be obtained independently of the season, which is of course for a breeding approach in poplar very promising. This system is therefore an interesting tool for the acceleration of tree breeding in genomics, but also biosafety research. We have done this work here at poplar, but one can think about to transfer the system to other tree species, but this has not been tested so far. Then we are checking the new breeding technique, including or using the mechanism of RNA interference, or short RNAi. The idea behind is that many pests like viruses, bacteria, fungi, and insects can cause severe damages in crop plants and reduce yield. Financial losses for farmers are dramatic, can up to more than 500 billion US dollars. In conventional agriculture, regular use of chemical pesticides are observed in organic farming. Also, they need something which has been approved as pesticide, but they also have to spray chemical. And these chemical pesticides can raise some problems because they have a rather broad spectrum of activity. As a result, it's not only the harmful organisms that are affected, but also other maybe useful organisms and insects. They can get into the groundwater via the rain, and the risk of resistance development in pathogens and pests are huge. And therefore, a possible alternative is to vaccinate plants with double-stranded RNA, which induce the RNAi mechanism. RNAi, or again RNA interference, is a natural system that cells use to regulate the activity of genes. The mechanism of post-transcriptional gene silencing is a mechanism of post-transcriptional gene silencing in eukaryotes, and it serves plants primarily as a natural defense mechanism against viruses and transposons. So, to introduce a little bit in DNA and RNA, DNA is, as you know, double-stranded. RNA normally is single-stranded when we refer to RNA or transfer RNA or to messenger RNA. Viruses have single-stranded, but also could have double-stranded RNA. When the single-stranded RNA enters the cell, the RNA-dependent RNA polymerase get active and forms double-stranded RNA. The presence of the double-stranded RNA in the cell alerts the immune system, including RNAi. The mechanism is very complex. It's shown by different arrows. So the presence of the mRNA is dismantled, which causes no translation of the mRNA and therefore also no protein biosynthesis. So the idea is now to use RNAi for biotechnological crop protection, because RNAi cannot be used only for or against viruses, but also are conceivable against fungi and insects. If a plant has a specific double-stranded RNA fragment that exactly matches to the mRNA of a pest, the mRNA of the pest is neutralized. In consequence, the protein is not formed, as I have shown before. And if the protein is vital for the pest, it dies or can no longer reproduce. In the US, Monsanto has produced the maize, MON 87411, which also includes the DVSNF7 double-stranded RNA. This is approved and in use, and it's grown on the farmer's field. It's a GMO. Okay, when think about the exogenous application of the double-stranded RNA. It can be either sprayed on leaves or directly on the insects. The double-stranded RNA can be uptaken by the pathogen. The very important point here is that is, at least in Europe, considered as a non-GMO approach, and therefore, because of the non-GMO approach, it's interesting for farmers but also foresters. Okay, this is that. The dsRNA is taken up by the harmful organism and then transported into the cells. The first product called Lectona, it's against the Colorado potato beetle, which is applied by sprays, is in the registration process in the US. The publication describing this work and this product is shown here. It's published in 2021 in Frontiers in Plant Science. And also further RNA sprays are being developed against, for instance, against the diamond bag moth or mold and varroa mite, which are parasitic on honeybees. Mold, for instance, high specificity, so only target pests are damaged. Non-target organisms are not affected. No residuous RNAi molecules are degraded very quickly. Unlike chemical pesticides, they do not accumulate in the environment. No development of resistances. Only spontaneous mutations in the sequence of the target gene could result in that the double-stranded RNA is not longer be active. And for our tests. And here on the right-hand side, you see again the work, the flow, starting with the gene target. No, the RNAi is induced, no mRNA is formed, no protein, and at the very end, the insect will be killed. Or thinking about why it's just a small effect and why not the double-stranded RNA has killed the larvae, and there are two possible explanations. One is that we have chosen the wrong genes, or the second possibility is that butterfly larvae are somehow tolerant against double-stranded RNA, and now we are going to beetles and check the same system on beetles, but this is work which is at present going on in our lab. Finally, coming to new breeding techniques, genome editing, CRISPR-Cas. And in our lab, this is the next slide, but just shortly introduce in CRISPR-Cas technology. It's fast and simple to use. It's a bacterial system which can be used when adapted also in eukaryotes. That's targeted mutagenesis. That is a very attractive feature. It's very highly precise. You can induce single nucleotide mutations, knockouts, deletions, and insertions. Mutations are not distinguishable to those obtained by spontaneous or X-ray or chemical mutagenesis, and breeding and domestication by using this technology can be accelerated in an undreamed manner. And now in our lab we have several applications which we has started. For instance, the full paralogs to increase biomass formation, which has not been published so far. We have tried to change mycorrhizal symbiosis by knocking out LysM CAR genes, which is leucine motif receptor-like kinases. We have knocked out the DOS19 gene, the tomosembrio defective. We have knocked out the tiller angel control gene, the TAC1, to induce horizontal growth. We have knocked out the ARR17 gene to induce a sex switch from female to male. And here you can see on this slide it was working very well. We have published this 2 years ago in Nature Plants with the first author Niels Müller. And on the picture you see we switched the sex from female to male by simply knocking out of this single gene. Another approach is using CRISPR-Cas ribonucleotides for mutagenesis, which is interesting because it's including no transfer of RNA. It's just a transient approach, an opposite to all of these. These are examples of trees which show a columnar, erect, or paramedalis habitus of growth, which could be interesting for short rotation coppices, because we had an idea when we used just three clones which show an erect or a columnar growth. The use of these trees could enable an increased number of plants per hectare in the field because of the narrow planting of the trees. The higher number of trees then should lead to a higher biomass production per hectare. Okay, so then we looked in the literature and found a paper on peach in which exactly this has been described, the different phenotypes. You can see here in the picture on the left hand slide, you find the standard peach tree, which do not show the columnar growth, or in this case it was called broomy or pillar, the erect form clone. The sequence is already available, and we designed guide RNAs for our genome editing approach. And in the slide, in the bottom you see with the red angles the positions where we try to induce the gene editing mutation. So I already mentioned that this inner clone has been used for transformations, 7171 before. We obtained it total 19 putative transgenic lines. Okay, for some lines we also isolated sublines. From PCR, we identified that from these 19, 11 were transgenic and 8 we found that found lines that were homozygous edited in both proteins, or homozygous edited, or heterozygous, just heterozygous edited in both proteins. But what is more important is the phenotype, and here is an view about, or in the in vitro, the tissue culture phenotype. On the left hand side you see a control plant with this white angel of here these petiole, leaf petioles. On the right hand side you see an edited, and there's an indication that the petiole angle is much smaller. Okay, this is in the in vitro. Then we planted the plants into the soil, and after 1 month growth in the glass house. When we have a second year plant on the left-hand side, or in the third year, now you can clearly see the erect growth of this poplar line. So after 3 years, now you see on the edited that we have induced the columnar growth in the 717 clone, Poplar canescens clone. We have used the for transformation. So in summary, these systems work. So to summarize all that, what I tried to introduce is that compared to crops, trees are poorly domesticated, thus the genetic gain could be very large. Tree improvement comprises tree form and growth, for instance biomass, and wood characteristics, for instance lignin. Classical tree breeding is slow due to T characteristics, longevity and extended vegetative periods. Tree breeding can be accelerated by a genetic engineering, high-speed breeding technology, and genome editing, but as well by genomic and marker approaches, which I have not shown here in this presentation because then it would be too long. And finally, RNAi is an interesting no GMO alternative